So hello to everyone, we are back in this other video. So for today we are going to observe how to modify a file within BiomiX, and I'll show you an example of metabolomics analysis using annotated file. So the first thing to do is open BiomiX. So I am in the folder containing BiomiX. Again, I'm working with Linux, so for Windows it's just enough to click in BiomiX executable file. Here instead we use the command line, so we click on it, and the first thing is upload the metadata. In this case we are going to use the cachexia metadata. Open. So to show you what is this cachexia metadata, actually is a very simple metadata including just the sample name and the condition, as you can see, cachexic and control. And what we are going to do is to show you how to modify a file. First of all, so you can see that, for instance, here in the metabolomics file, in the input, you have this type of file. As you can see, there is a little problem. Sorry, there is a little problem, because if you see here what's happened, we have this column condition cachexic, the ID including the sample name, and then the metabolomics data, the metabolomics variable. What's the problem? The problem is that the format that is asked in BiomiX is always the same, and it includes the name of the sample, so this one, in the column, and the name of the variable in the rows, and there should not be any column called condition. That is the situation that the majority of you can actually meet in the reality, and that's why BiomiX has been made to solve this type of problem. What's happened? How can we fix this dataset without going to our analysis? To do that, the first thing to do is actually upload the file. So as you can see, metabolomics input, we are going to click on compounds name. This one is the one already modified. But as you can see, you can open it, and it's going to be asked, do you want to modify the file? Here normally you select no and the file is uploaded, but in this case we are going to click yes. What is going to be asked is which is the separator in your data. To do that, you need just to open it in a text editor to see, and as you can see here, this space is a tab. So what you need to do is just where is the main interface? Here. You need just to select tab. Okay. The next question is, does the dataset have an header? In this case, we are going to switch the column name with the sample name. So in this case, just for the sake, we will write no. Otherwise, we are going to lose the header if we select yes. What's going to happen now is that we have the matrix. We have the matrix including including the samples. Of course, that is just the first 10 lines and the first 10 columns. if we, in this preview interface, if we click on tab, it's possible to transpose the matrix. It's possible to remove rows, remove columns, and then save the matrix. So the first thing that we are going to do is remove the second column because it is not important for us. So we are going to click on remove columns, we are going to click on second columns and click OK. And now the preview will be regenerated automatically. And as you can see now, the second column is disappeared. What we need to do now is to transpose these columns in the other side and the metabolomic variable in this side. To do that, we need to click on transpose. As you can see now, we have the matrix with the right format. And the only thing that we have to do is save matrix. We will call it OK, the same with OK. What we have to do, just in this case at least, we can open it in editor text, and as you can see, you can observe that the format is right. There is just the rows, the header that must be removed. In this case, save it, and here you have the right format for the analysis. So the only thing that you need to do now is upload the file directly here. Compounds name path. You want to modify the file? This time no, because it's already modified. And then select metabolomics analysis, so you want to analyze it. The control must be control versus cachexic, and the output must be saved in example folder that is here. We don't want the integration. For the advanced option, we have to click that this dataset is already annotated, and the name is the compound name. To conclude, there is nothing more to select. We can save, and we can start the analysis. Sorry, I actually forgot about adding plasma in the label. So I restart the analysis, and as you can see, the analysis started. So here we have all the samples. We have information about this distribution, the normal distribution, if our variable follow a normal distribution or not. And the analysis is over. So what you can see here is that as output we can observe plasma cachexic, and you have the heat map with all the values, not clustering a lot at least, and the volcano plot having just positive values. And information about the metabolites upregulated here with all the p-value, log to full change, p-adjusted, and names. You have peak statistics, so just name, log to full change, p-adjusted, p-value. Maybe it's better to visualize it like that. Yeah. And the metadata, of course, and also a section with the MetaboAnalyst. Here you have files that can be automatically uploaded in MetaboAnalyst. in this case there is no much. There is, for instance, the log to fold change for each metabolite that you can just upload directly in MetaboAnalyst to analyze them in each section, like joint pathway analysis, in network analysis, and in pathway analysis. So you need just to copy and paste like I did in HR. Wait, I show you. So just to show you something that you can do, you can click, for instance, in network analysis here. Is explained in the website that actually for the network analysis and the joint pathway analysis, what's happening is that there are the analysis with the metabolomics with the result of your sample, but what's very nice is that if exists the same dataset having the same label, so if you have I don't know, cachexic, including also other datasets like transcriptomics in particular, BiomiX is automatically able to save the metabolomics result, but also the transcriptomics result coming from other sample. Just imagine now, I don't know, and whole blood RNA-seq in cachexic sample, in the case there will be, is able to use the transcriptomics result and upload it automatically in together with the metabolomics one to do an analysis, a joint analysis. With the network analysis, for instance, here you have, if you open it, you have just the, how to say, the metabolomics result, but in the case there will be also transcriptomics file, there will be also a file including transcriptomics here. And why that is important? Because if you open it and you use the network analysis in MetaboAnalyst, you can obtain a very nice, very cool, let's say, very cool pathways and network analysis. So you can copy that, you can select, click here to start. Here, for instance, network analysis. As you can see here, you can upload the genes, and here you can upload the metabolomics. So we have compounds name, we copy it. In the case it will be also transcriptomics, you can add the second column and then submit. Takes some minutes, and wait. Okay, it's over. So you have the metabolites that you upload, and we click on submit, and here we can select different type of how to say, network that we want to explore. For instance, there is the KEG1, where we can observe the different metabolites within the huge network, metabolomics network. So it's a very nice tool. To explore. You can of course go deep and see all the network, and there is also the metabolite interaction, but also the gene metabolite network. So for real, this allows you to do whatever you want, to explore the data in a very nice way. As you can see, with expression and the values. Okay, so that has been an example. I hope it was kind of nice. The result, I show you the results. So I stop 1 second. Okay, so for the next section we'll continue with the metabolomics, and this time we are going to analyze using the MS2. So running again the same code, this time we are going to analyze some data from MS2. So again with the dataset of autoimmune disease. And what we are going to do is selecting again the same disease, comparing lupus and control, selecting the output, so example here. And what we are doing now is actually show you how to how does it work an MS2 analysis on BiomiX with metabolomics clearly. So what we can start is, of course, clicking on metabolomics here in the first input, so filling the first input. So we are clicking here. We are not going to integrate anything, and we will call it plasma. What we are going to do now is upload the metadata. Sorry, the file. So we are going to upload the matrix. In particular, where is plasma all? We want to undefine? No. So just to show you the data set quickly, is exactly the same format as always. So sample in the columns and rows in and variables in the row. So it's where plasma all. As you can see, sample in the column and variables in the rows. But as you can see, this time we don't have metabolites name. We don't have human metabolites name or compounds name. But here we have just peaks. So we don't know exactly who is what's happening. Who is behind this specific peak. If you remember from other dataset like the one that we used last time, instead here we have names. We can also use in BiomiX dataset including this one, having the human metabolome number that is exactly corresponding to a specific metabolite. But when we have just peak, we need to find the annotation for this specific peak, and that's why we do an MS1 or MS2 annotation. What's the difference? The difference is that, as I explained you, MS1 is just using information, annotation information, like this one that I'm going to show you, including the peaks that you have, and mass charge ratio, like this one, so including the ratio between the mass and the charge of this specific metabolite, the retention time that can be in seconds or in minutes, or both of them. These are all the information required for an MS1 annotation. For the MS2, we need something called MZ files. These MZ files contains the fragmentation spectra. What's the fragmentation spectra? If you write online, you have something like that. The fragmentation spectra is literally the signals coming from the fragmentation of the original molecule, of the original metabolite, giving a fingerprint unique for each metabolite. And so these files contains this specific fingerprint. Continuing now, what we need to do in practice for an MS2 annotation is click on the advanced option, go in metabolomics annotation, select MS1 and MS2 annotation, and then add the MS1 annotation that is in input 1. So we are going to select plasma annotation, and then select the MS2 annotation that is inside the MZ files. So we just select the directory, and then having, as we have plasma, is going to use the MZ file, MZML file, having plasma. We can add more than one, of course. The important is providing the sequential number for each of them. We don't need anything else, I think, and we can start the analysis. Is retrieving here from a human metabolome dataset, and I think I'm going to stop the video for to save time, but the analysis is start, so it's starting the annotation with MS1 at first and then MS2. So just a little update. As you can see, here we have the analysis that has been continued. Now I started to read the MS2 files. As you can see, it's proceeding with the reading, and he found 176 peaks out of 500, 301. So that means that this peaks contains also fragmentation spectra, and these one are going to be annotated within the 531. So it's using, in this case, 3 different datasets, because I forgot to mention that if you go in the advanced option, now it's blocked, you can select different dataset. You can select the human metabolome database, the MONA, and the mass bank. So based on the priority that you will provide, it's going to use all of them in a different order, or just one of them based on your desire. So just a little point. So again, a little update. As you can see in this part, is actually saving the MS2 spectrum, so it's possible to observe this the spectra directly in the output. And in this part, instead, is connecting to SEOMAS mediator, the repository or line for the MS1 files, MS1 annotation, and is then asking to the server to connect to find the match with the MS1. So again, the total analysis lasts more or less 10 minutes, so that's why I'm saving time cutting the video. Here, little update has been this is the analysis with the SEOMAS mediator. As you can see, it was uploading the files. After this part, what's happening is providing you the peaks that are not annotated. So using the spectra, actually there was no match, and that's why they are listed here. What else? Here there is the part in which starts the biological pathway analysis. So it's using MetPath to detect biological pathways if they exist, if they are significant also. Okay, finally the analysis is finished. So we'll go through the output just to show you the results. Plasma lupus. Okay, here, as you can see, you have the same results as usual. So you have the heat map for the condition. You have the differential expressed molecules here, and it's the same information that you observe also for the metabolomics analysis with the annotation. But the thing that I want to show you now here, something more, let's say, is the spectra. So for each dataset here, you can explore each spectra for each annotated peak. So if, for instance, you are wondering how the annotation of the peak 1 has been made, if for the specific metabolite is a good or not the spectra, you can actually click on it and observe how the spectra has been matched, in which site. I think, for instance, here we have the valine. So here is the database MS2, and this one is the spectra that you obtained from your dataset. As you can see, the annotation of the valine has been obtained because we have these 3 sites in which the spectra match. So that's all for the metabolomics part. So see you in the next section.